Hello, let's talk about the Bio 113 lab kit and how to approach this semester. Now, this kitty who's going to help us with our lab kit today and understand what we're doing loves paper bags. So we have this classy paper bag that you will be handed at the when you come to pick up your lab kit. Now, one of the things that's going to be in your lab kit is your lab manual. Unfortunately, I had to make revisions for our class and have those lab manuals printed long in advance of our class starting. And unfortunately, the administration didn't give notice of the delayed start until December 22nd. And by that point, those lab manuals had already been printed. So what you're going to see in the email that includes the link to this video is the revised protocols for the grass lab, and I had to make those revisions because the notification of the delayed start occurred after I already had the lab manuals sent to the printers, and they were finished printing. So please note that in your lab manual for the grass lab, you do have the readings, and the readings are still the same, but the protocols, how we're going to conduct our experiments, will change. In the lab manual that you're receiving in your lab kit, you will have the lab report, a place to note down some thoughts, and then also record your data. Because of COVID, because we're trying to limit the amount of interaction or potential cross-contamination, we will not be turning in any physical lab copies this semester. So while your lab manual does have lab reports, what you're going to find is that all of those questions and everything is online for you to submit. So please don't try to staple lab reports together and turn them in. That's just for those of you who need that hard copy and want to be able to write things down as you are conducting the lab that you will then enter electronically. So in your lab kit, you are going to have your lab manual. The other thing that you're going to have in your lab kit is you're going to have the FAST plants. Now your FAST plants are going to really be a FAST plant kit. In the FAST plant kit, you have everything you need. You're going to have your large and your small deli container. The small deli container will already have some soil, and that soil will be pre-wetted. What you're going to do is remove the lid from that soil. The soil already has the wick to connect the small container to the large container. You're going to remove the lid, take your FAST plant seeds, plant the 10 seeds in that circle pattern in your FAST plant small deli container with soil. You're going to replace some of that soil or sprinkle some of the soil over the top, and then you're going to get the surface super wet. When our seeds start to grow, they don't actually need sunlight. What they need is to stay consistently and continually wet. The water flowing into the seed is what triggers germination, triggers that first root to start to come out, and then those two little seed leaves, the two little cotyledons, will start to rise as well. Included in your FAST plant kit will be a little set of directions, and in those directions I give you some tips to watch for, because your seeds are going to germinate within 1 to 2 days, and very shortly after those cotyledon seed leaves poke out of the surface of the soil, you could run into this problem of this hypocotyl, the in-between section between the cotyledons and the soil. This hypocotyl start to stretch and elongate. And the problem with that is that it's very weak, and we need that section to stay short. We need it to stay very strong to support the developing leaves and all of the weight of that mature plant when it gets to that point. The other thing that can happen is as your hypocotyl elongates, it may start to bend, and that's essentially the plant trying to reach out for the light and reach for that food source. So make sure to read through those directions that I have for what to look for and what to keep track of in the first couple of days as your baby plants are starting to grow. Just like any other baby, a baby kitten, a baby puppy, or a baby human, those first initial stages are so crucial and really do set the course for the lifespan of the adult plant. So we want to make sure that we do everything right in those first couple of days, because if anything happens, we won't be able to correct that. So complete your fast plant homework, watch the video, and then plant your fast plants as soon as you possibly can. You have a fast plant journal that is due on Saturday. So let's say this week has been crazy. You've been working like nuts. You don't have a second to do the homework and then work with the fast plant and kit and get those fast plants planted. That's okay. If you start Saturday and you still need to turn in your plant journal by 11:59 p.m. Saturday night, so you're going to have to write about what you expect is happening, what you hope is happening, or what you think will happen in the next couple of days. One of the things to keep in mind with the Fast Plant Journal, always use terminology. So what you'll see in the introductory slides is that I start to introduce some terms, I start to talk a little bit about different concepts, and then even in your Fast Plant directions and the Fast Plant homework, we have some terminology like hypocotyl, cotyledon, first true root. I want to see those types of terms show up in your plant journal. Your plant journal is Fast Plant Journal is meant to reinforce what you are learning in lecture, reinforce these concepts. It's another way for you to practice things. So, you get your lab kit, you tear open your lab manual because you want to see the pretty pictures and see what I'm babbling about the grass lab and protocols being weird. You get your Fast Plant kit, you open that up, you get your Fast Plants started. You can complete that Fast Plant Journal at any time, but I have to be pretty strict on the due dates. So let's say you're super motivated. You get your lab kit on Tuesday, and by Tuesday night, you have planted your fast plants, and you want to do that journal on Wednesday morning. Rock on. I love it. Try to be consistent when you're doing your journals. Try to then do it every Wednesday morning, so that we have that kind of consistent tracking of our fast plants over time. If you need any help putting photos into your Fast Plant Journal, don't hesitate to email me, or better yet, drop into the Zoom office hours, or even the Zoom office hours, because I can usually explain how to upload photos into the Fast Plant Journal in about 2 minutes. So it's not a really long thing to figure out how to do that. So you've got your lab manual, you've got your Fast Plant kit. The third thing that you're going to find in your lab kit is going to be the supplies for the grass lab. So for exercise A of our grass lab, what you're going to be doing is you are going to be dissecting a little baby corn plant. Now, in order to do this, your corn plant is going to be planted in some soil. So if you can't do this right away after getting your lab kit, either Wednesday morning or Thursday, make sure that you're attending to that grass plant and that you keep it alive so that you can do the dissection with a living plant and not a plant that's either dying or already dead. So you'll have your grass plant growing in some soil, and what you're going to do is you're going to gently pull it out, and then you're going to lay it on a cutting board, and you're going to dissect it. I'm going to have a video that goes along with this, but like you, I have to wait to get into campus. There are all these different permission slips. So as soon as I get my corn plant, I will be uploading a video. Where you are going to find that video, go to the Eagle course site, click on modules. When you look under the Unit 1 heading, scroll all the way down to where it says Grass Lab Report. And under Grass Lab Report, you're going to see Exercise A, How to Dissect the Corn Plant video. So I will have a video showing you how to dissect that little corn plant. Just please make sure to keep it alive. Give it a little bit of sunlight, give it a little bit of water. Don't let it get too cold. It is corn until you're ready to dissect it. The other thing that you're going to find is you're going to find the supplies for Exercise B. Now for Exercise B, we're going to be making a gluten ball. It is absolutely crucial that you watch the video before you start to do the experiment, before you start to do the exercise. So in our lab, we have lots of different types of flour that we are going to be using. And the flour is, all of our flour that we're using for this class comes from grass plants. Now, some of the grass plants are wheat, some of the grass plants are barley, some of it's oat, but everything that we're using is going to be a grass plant. Some of the grass plants that we're using, the flour that we're using is really, really hard to track down and is kind of rare. So the einkorn flour and the red turkey wheat that's new this semester, those are kind of specialty flours that I was able to find for us to use. So you pull out your bag. I don't know which kind of flour type you're going to get. It's random. But you will have one type of flour that's in the list in your lab manual. It's already pre-measured. We've got 25 grams of flour, and you'll have a piece of cheesecloth. Once you watch the video, you will know the techniques. Now, the one thing that I want to highlight here is please, please, please be careful when you're adding water. Some of our types of flour, if you add water, it'll go from consistent, cohesive little ball to complete sloppy mess in an instant. So you want to add the water slowly, stirring and mixing. You want everything to come together to make a nice cohesive ball, like a bouncy ball. Be very, very careful. Now, I know that mistakes happen. That's the joy of experimentation. Don't feel bad. Just go into your cupboard. Hopefully you have some flour. You can use any type of flour that you have. You probably have some all-purpose flour. Measure out 25 grams, and you can repeat the experiment again. Ideally, nobody makes a mistake, and we all get data from the different gluten balls so that we can see, look for some trends in our grass lab. Look for some trends in the grass flour that we are using and is commonly used for baking bread. So you will make your gluten ball, and the one thing that I want to point out is when you're making your gluten ball in the video, I talk about bringing your gluten ball into class. Now the video was made for last fall's lab when we had the tornado come through and our lab was delayed. We were able to get in and the timing worked out differently. This year you're going to need to weigh your gluten ball at home. So that's really the only difference between the video and what we need to do this. Now hopefully you have a kitchen scale at home, but if you don't have a kitchen or a food scale at home, that's okay. This is what they look like. I think that this scale cost us all of $10. They're fairly cheap. I don't want you to spend a tremendous amount of money if you're going to buy one. This has lasted us for many, many years, so they're pretty durable little things. They're easy to operate. In order to turn it on, I hit this button over here. You'll also notice that that button says tare. And what this is going to do is allow us to be able to subtract the weight of whatever vessel we're using to measure. So in the case of measuring out this flour in the plastic bag, I first put the plastic bag on the scale, hit that tare, subtracted the weight of the plastic bag, and then I could then I could put the flour into the bag and get an accurate weight. This one automatically records in grams. So if you don't have a kitchen scale at home, you're not able to purchase a kitchen scale, please email me as soon as possible, and we'll work out some kind of arrangement to make sure that you can get your dried gluten ball weighed and you can get that data sent to me as soon as possible. So that is exercise B, making gluten ball. The other supply that you're going to have in your lab kit will be the supplies for baking bread. The same flour that you used for exercise B to make the gluten balls will be the type of flour that you use for baking bread. I don't know your experience if you've baked bread before. If you haven't, or even if you have, I highly, highly, highly suggest watching the video. Remember, videos are found on the Eagle course site. Click on modules, look under Unit 1 heading, and scroll down to the Grass Lab report, and you will find that exercise C baking bread video. For our bread recipe, you are going to have a pre-measured baggie of 1.25 cups of flour. Again, it's the same flour that you used for exercise B, so keeping with your experience, helping you to get a lot of experience with one type of grass and the flour made from that seed, or we could consider it the grain fruit. So you have your pre-measured flour. You'll have yeast in a packet similar to this, and you will have 2 tablespoons of sugar. All the rest of the supplies, hopefully you have in your kitchen. You're going to be measuring your dough before it rises and then after it rises. This is going to help us to understand sort of the durability or the strength of the flour type that you're using. It's going to help us to understand the role of protein in flour as far as how the bread tastes, the texture of the bread, and why we prefer maybe one type of grass flour over another type of grass flour. So make sure you get that measurement before and make sure you get that measurement after. And then, of course, you're going to be baking your bread. Let us know how it tastes. Hopefully it tastes delicious. Now, what you'll notice in the syllabus is that I'm asking for all data from exercise B and exercise C to be emailed to me by January 28th. Now, I don't care if you send it to me earlier. I hope you will. That will help ease my own personal anxiety because I'm going to be collecting everybody's data. The sooner I get everybody's data, the sooner I can send it out to the whole class and everybody can get to graphing that data and looking for the trends. The other thing that I want to let you know is that sometimes the trends are not pretty. We would love to see a really strong correlation between the amount of protein and the amount of rise that we get in our bread dough. More protein, more rise in our bread dough. More protein, the dough is less sticky. The dough is more elastic. In other words, I press it with my finger and it bounces back and makes that beautiful smooth shape. That's what we hope would happen. But unfortunately, that isn't always the case. And part of that does have to do with the idea that a lot of us are not experts in bread making. This might be our first attempt. We aren't familiar with the experiments, and so there's a lot of uncertainty. That's all okay. I'm okay if the data isn't pretty. I'm okay if the trends aren't perfect. This is actually more accurate to how science looks and how it works and how it feels, especially when you're conducting experiments with other scientists and other researchers. So, do the best that you can with the interpretation. Know that we've got the ideal, what we wish would happen, but sometimes our data doesn't always look that way. Sometimes we just have to report what it is we are seeing in the data. And that exercise is far more valuable than if everything works perfect and everything turns out as expected. So in our first labs, the lab number 1 is a combination of the scientific method review, which hopefully you've had before, and then we use a microscope. Ideally, we would start this on the first day of class, complete it, and then we would move on to our grasses, starting to get into more complicated experiments. But unfortunately, we can't do that this semester. So for lab number 1, on the scientific method, in your lab manual, it's got the readings, and the questions are on Eagle. Please complete exercise A of lab 1 as soon as possible whenever you get a chance. That will make your life a little bit easier. When we do meet together on our first lab class on February 2nd, it's going to be busy. We're going to have to finish lab 1 using a microscope and making those microscopic observations. And then we also have to jump into the legume lab, and we're going to do the entire legume lab our first time together. I hate this. Usually by this point, we would have had some time working together. We would have built a relationship. We would have gotten familiar with things. But because of this delay, we're going to have to jump into the deep end. I want you to be prepared. So, you get your lab kit, you plant your fast plants. You look at lab 1, you start to work on exercise A and get that scientific method, get those questions answered on Eagle, get it out of the way. You don't have to complete the whole lab. You won't be able to for lab 1. But you can get those questions answered. Eagle will save those answers. Get it out of your way. Then jump into the grass lab. Exercise A, do your plant dissection. It's going to be fast. Answer those questions on Eagle. Then do exercise B. Make your gluten ball. Get that data, send it to me. Then do exercise C. Get that data, send it to me. When you look at the syllabus, it clearly lays out what I hope you're doing each week so that you don't feel overwhelmed. In the lab manual, you're going to see for the grass lab, exercise D. We don't have the supplies for that in your kit. That's something that the lab technician and I have done. We've taken the photographs and we've just integrated it into the lab report, so you will see that posted in the lab report. So don't worry about the last part of the grass lab. That's already been done for you. Then when we meet, we're going to do our legume lab and finish up lab 1, the microscope component. So it will be a busy first day when we are in lab together. As always, email me, drop into those Zoom office hours. Please don't agonize if you're having any troubles. The nice thing about technology is that you could take a video of your bread. You could take a video or a picture of your gluten ball and send it to me and say, I don't know, does this look okay? Did I do this correctly? And I can so quickly assess and send you back information. So please don't agonize over anything. If you have any problems, just email me. Please send me pictures, send me videos. I want to be able to help as much as I can. Thank you so much for your flexibility. Thank you for adapting to all of these changing circumstances. I wish I could tell you that you won't have to keep doing this for the rest of your life. But the reality is that this is how life goes. We often have to keep adjusting. And it is our capability to be flexible in the face of adversity, in the face of changes. That is what really makes a difference. So, you got your lab kit, you're starting out and you're working through this. That is huge. Give yourself credit for making those first steps. In 16 weeks, we can look back and reflect with a big sigh of relief, and hopefully everything goes well. As always, email me if you have any questions, and I hope you have fun. These first couple of labs, especially baking the bread, can be really